Please welcome Jin-San Yu, CEO and President of Pharmapsin, which specializes in the development of next-generation antibody therapeutics.
Thank you, organizer. Thank you, chairman. And thank you, audience who take the time to listen Pharmapsin's presentation. We have a website called pharmapsin.com. If you need more information, please visit our website. And there is also my email address, jinsan.yu@pharmapsin.com, or simply you can send me email to wincancer@gmail.com. I'm going to start with the take-home message of my today's presentation. First, the olinbasmab. Olinbasmab is anti-human VEGFR2 neutralizing antibody. And I'm very excited to share our current status. So Pharmapsin and Merck are doing clinical trial for olinbasmab with Keytruda combo for metastatic triple-negative breast cancer. And this phase 2 study will open very soon in Australia. And olinbasmab Keytruda combo for mTNBC phase 1b is still ongoing and deliver 50% ORR and 67% TCR with clear safety profile. And some patients are still in PR, although the study length is already an end, but we are happy to providing our all necessary supplies for those patients as a compassionate use. And both Pharmapsin and Merck have filed their patent of olinbasmab plus Keytruda combo very recently. And olinbasmab plus Keytruda combo for recurrent GBM phase 1b goes still smoothly. And the first cohort of olinbasmab monotherapy, which is 60 mg per kg biweekly for Avastin non-responding recurrent GBM phase 2 in the States and Australia is completed. And we open the second cohort, which means 20 mg per kg biweekly dose. And currently we have 2 patients already in our studies. And combination of olinbasmab with various modalities are in preparation for different indications. I'm going to introduce also our another asset called PMC309, which is anti-VISTA antagonizing antibody, which has a nanomolar affinity with highest specificity to VISTA. And PMC309 has shown anti-tumor effects in various animal model systems. And interestingly, and also as expected, combination with anti-PD-1 combo has shown synergistic effect like other competitors. And our PMC309 uniquely block VISTA-VISTA interaction, VISTA-V6-3 interaction, and VISTA-PSCL1 interaction in various pH environments. So what we are looking for in this convention, we are looking for funding about or more than $50 million for strategic investors or financial investors for our several interesting clinical trials. And we welcome our new shareholders. We would like to expose potential shareholders abroad. And we are looking for opportunity of licensing out for our clinical and non-clinical assets. Co-development partner is needed for olinbasmab and olinbasmab combo trials, and PMC309, PMC42, PMC43, and other early assets. And we are also open to discuss potential joint venture formations. So who we are actually, Pharmapsin was founded on September 3rd, 2008 by engagement from Novartis and OrbiMed. And we are mainly focusing on next generation antibody therapeutics. We are listed on Kosdaq, and the market cap is around $200 million is fluctuate at the moment. And we are clinical stage with extensive experience and innovative platforms and competitive assets. And as I mentioned, olinbasimab, in the beginning, we thought it is kind of just tumor angiogenesis inhibitor, but the more we study and experience with olinbasimab and trial, olinbasimab plays as an immune modulator in tumor microenvironment with clear safety profile again. And we also have 3 first-in-class oncology and non-oncology molecules being prepared for the clinical stage in next years. So we differentiate from other startups, as I mentioned. It was engagement from Novartis and OrbiMed. We have one of the best fully human antibody library called QPhage display library with 10 to 11 diversity. We have several antibody-based assets, but also bispecific and multispecific antibody platform and their pipelines. We have a strong relationship, partnership with Merck, Sanofi, and Roche. We have a strong relationship with world-class hospitals and experts. And we have great talented pool from scientific advisory board and consultant and management. So in the beginning, we started with tumor angiogenesis inhibitor, but now we expand the pipeline paradigm to vessel normalizers in oncology, but also vessel normalized for non-oncology asset. And we are expanding also immuno-oncology as first VISTA, but we have several other bispecific antibody playing in immuno-oncology assets. We have a US subsidiary called Winkel Biopharm located in South San Francisco. And they are mainly focusing on either formulation for AMD and DR, which is currently intravitreal injections. And they are also focusing on some pipeline for aneurysm and other vascular related diseases. So this slide shows the overview of pipeline. As I mentioned, olinbasimab pembro is in mTNBC and recurrent GBM and ovarian refractory recurrent GBM. But anti-VISTA, we are focused on solid tumors, PMC4/3 as a type 2 activator and vessel normalizer. We are focusing on ocular and solid tumors. And under the collaboration with NIH, we are also focusing on SCLS, which is Claxon disease. And early stage asset, we have those assets, which is at the moment interesting is PMC122 targeting PD-L1 and CD47, mainly enriching on tumor microenvironment and minimize the binding on CD47 on red blood cell to maintain the better safety profile. We have anti-EGFR-V3 specific antibody, which can be applied for CAR-T, CAR-NK, and other modalities. So as I mentioned already, olinbasimab is very interesting molecule, very safe, but also uniquely efficacious. Just one example, both Avastin and Cyramza they have failed in HER2-negative breast cancer. Olinbasimab performs very well in HER2-negative or triple-negative breast cancers. And even Cyramza failed in recurrent GBM. We have quite promising clinical data of olinbasimab alone or through the combo in recurrent GBM too. So this is the competitive landscape. Avastin is the landmark in this mode of action. They have also biosimilars. But VEGFR2 targeting Cyramza is marketed for gastric and other solid tumors, and Geltrap2. But they have all common side effects, which is hypertension, gastric lung perforation, hemorrhage, and proteinuria, whereas we never observed those side effects by olinvestmab. What we observe is reversible capillary hemangioma, mostly grade 1, sometimes grade 2 on the skin, but not those common side effects caused by VEGF-R2 antagonist molecules. So we are very excited about the progress of olinvestmab with Keytruda in metastatic triple-negative breast cancer, and we will see the phase 2 with higher number of patients, whether we can reconfirm and reproduce the data we observe. So again, PMC 309, the VISTA antibody, is kind of next generation from anti-PD-1 or anti-PD-L1 or anti-CTLA-4. It has a huge biology on MDSC and maintain immunosuppressive environment of tumor microenvironment. And also it has a role to activate the regulatory T cells, but blocking the VISTA by PMC309, we can reduce the biology of MDSC and activate the T cells. And we have shown several solid tumor animal model system the anti-tumor effect. And if you combine with anti-PD-1 antibody, it's quite synergistic. And we're gonna enter phase 1 next year in Australia. So this is the mode of action of PMC309. And we presented at AACR this year and recently CASMO, and our poster presentation of PMC309 was awarded the best poster presentations. Very specific compared to competitors, and that probably support a better safety profile, which we already observed one of the anti-VISTA antibody in phase 1 by J&J. They observed CRS, and we think their antibody has some specificity issues. And we nicely observed that it is able to activate T cells and inactivate the MDSC same time. And again, combo with anti-PD-1 is always synergistic, regardless of solid tumor types. As I mentioned, we are heading to phase 1 next year, but Curis has already initiated their phase 1, and PFAVR initiated phase 1. But besides these 2 assets, all other competitors are same stage, and therefore we are not far away from the first runners. PMC43 is very interesting molecule. It binds Tie2 with nanomolar affinity. And what it does is remarkable. It simply renormalize, reorganize, disorganize leaky pathological vessel to normal healthy vessels. And because of that, it has a wide expandability of various indications, not only in oncology, but more likely several non-oncology fields. We have extensive research collaboration with NIH under the MCADA. There's a PI named Kirk, who's a world famous scientist with strong experience with systemic capillary leak syndrome. Clarkson's disease is another name. And the research collaboration and the results are very exciting. And I strongly hope we can share the results next time. And again, PMC43 is ready for file IND next year and enter phase 1. And we thought that we are the first one, but there are some other players who try to activate TIE2 and renormalize pathological vessels. So this mode of action is well known, and we hope that we can utilize for also ophthalmology field. That's why we presented ARVO 2021 with our data. And as you can see, our PMP43, the TIE2 activator, has a strong biology in vessel normalization and also reduce the level of androgenous regulations. And also PMP43, similar to Eylea, it has value to repair the leakiness of leaky vessels. However, more exciting for PMP43 is it has a better performing in terms of optic nerve responses recovery compared to Eylea. So this area is quite crowded, but we are very excited with the result of PMP43. And beside oncology and ophthalmology, we are exploring, other SCLS, but also we're looking at the diabetic related kidney disease. And we are also planning for COVID-19 with ARDS and some other disease which is related to non-functional or pathological vessel issues. Another asset, PMP42, has also very strong biology to renormalize pathological vessels and also target the same target, but the epitope is different and the biology is different. So we are also exploring and developing PMP42. So that is the end of my presentation. And again, if you have any questions, visit our website or send me email simply, and then we can discuss in more details. Thank you very much.